Okay, this is this second recording for the Chapter 38 slides. And I just, even though we already passed this, I want to review the parts of the eye, and then I want to review how light is, how the light receptors, the photoreceptors of the eye detect light, and then that is transmitted to an image. Impulses are sent to the brain, and then we see that the light as an image. So basically, the parts of the eye, one more time, that very outer layer is going to be really hard. This is going to be a really hard, tough structure whenever we do this dissection of the eye. That outer layer is the sclera, and then again, the cornea is basically is continuous with the sclera. So the cornea and then the sclera are the same layer, but the cornea is just the part at the front of the eye, the part that we see, except what's covered up, by skin, by our eyelids. Then we have the colored portion of the eye, which is the iris, and then there is a hole in the middle of that colored portion, and it's called the pupil. And here is the lens connected by suspensory ligaments to the ciliary body, and that ciliary body is going to contain the ciliary muscles, which control the size of the lens. The middle layer of the eye is called the choroid, and then the inner layer is called the retina. So the fluid in the front here is called the aqueous humor, and then in this posterior compartment is called the vitreous humor. So light is going to pass through the pupil and through the lens, and then it's going to be focused on the back of the eye, which is called the retina. And once the light hits the retina, that light is going to be the first the photoreceptors that are going to detect the light that comes in are going to be the rods and the cones. And just remember, the rod cells are for black and white vision, and the cone cells are for color vision and fine detail. So they are going to contain the pigment rhodopsin, and they're going to they are the sensory receptors for light, called photoreceptors. So the rods and cones will send that signal. Once they receive the light, they will send that signal to bipolar cells, and then those bipolar cells will transmit the signal to ganglion cells, which will transmit the signal to the optic nerve, which will take the signal to the thalamus in the brain, the thalamus, and then to the visual association area in the occipital lobe of the cerebral cortex. Now, some disorders of vision. Diabetics can have a disorder called diabetic retinopathy. So if you are diabetic, and I am, and my daughter is, we have to have a vision exam every single year, and they always check our retina, because sometimes, the eye doctors told me this many times, we can often detect problems with your diabetes. When your diabetes has become out of control, we can detect that problem first, because we'll see it in the capillaries. We'll see damage to the capillaries of your retina. So they're looking at the little tiny blood vessels called capillaries, and they're looking for damage. And that will be caused by your blood glucose levels not being managed. So that's one condition diabetics can get. Macular degeneration is also starts with the capillaries. It's when, in fact, diabetic retinopathy can lead to macular degeneration. When the capillaries supplying the retinas become damaged, this can cause hemorrhages and blocked vessels, and it is the leading cause of blindness for people in the United States under 65 years old. It's called macular degeneration. And then there is retinal detachment, which is when the retina peels away from the choroid layer, and it can be reattached with laser surgery. So you hear about LASIK surgery, people having LASIK surgery. We saw the optic chiasma when we dissected the sheep brain, but a lot of us, including myself, it was easy to cut off the branches of the optic nerve. They cross here at the optic chiasma. So from the optic nerve, the nerve impulses from your right eye cross over to the left hemisphere of your brain, to that visual region first the thalamus, and then the visual area in the occipital lobe of the cerebral cortex. And then from your left eye, they cross over to the right hemisphere. So the optic chiasma is that place where the optic nerve from your right and left eye crosses and goes to the opposite hemispheres in the brain. Now, glaucoma is when the drainage system of the eyes fail, and that can lead to a buildup of aqueous humor and increased pressure inside the eye, intraocular pressure. So basically it comes from a buildup of aqueous humor. That was the fluid that forms between the cornea and the lens, basically, or the cornea and your iris. And then you can have cataracts, and that's when the lens becomes opaque. It makes it hard to see. When it's opaque, it's incapable of transmitting light rays, and that tends to happen more in older people. And then this can happen with aging, or it can just be something that you're born with, but when we use the term myopic, we mean nearsighted. That means that I always forget. Hold on. Okay, so if you have nearsightedness, that means that you have a long eye, which means the rays focus in front of the retina when viewing distant objects. So in other words, when you're nearsighted, you can see close up, but you have a hard time seeing far away. When you're farsighted, you can see far away, but you have problems seeing close up. And then a lot of people, most everybody has some astigmatism, some degree of astigmatism, which is just fuzziness. So anyway, nearsighted means that you must wear concave lenses. That means your lenses have to bulge out instead of forming a depression. So nearsightedness comes from elongated eye. That means you can see better close up, but not as good far away. So your lenses have to be concave, or they have to bulge out instead of having a convex means that they I'm saying that opposite. Sorry. Concave means they have a depression, and convex means that they bulge out. I'm so sorry. Okay. Farsighted means you see better far away. That's hyperopic, and that comes from having a shortened eye. And you must wear convex lenses, which convex is when the lens bulges out, and concave is when it has a depression in it. All right. So nearsighted, concave lenses, farsighted, convex lenses. And then astigmatism is when you have like a fuzzy image, when you have your vision is kind of blurry, and everybody has a little bit of that. That comes from an uneven cornea or lens, and sometimes that requires glasses and sometimes it doesn't. All right, now we're going to move on to, that was the human eye, so now the human ear. The human ear, let's look at the illustration first. First, we have the outer ear, and the outer ear consists of the pinna, which is this portion, the upper portion, the earlobe, and then the auditory canal. And then the middle ear consists of, this is what we call the eardrum, but it's the tympanic membrane. And then the bones of the middle ear are called the malleus, the incus, and the stapes. Malleus, incus, stapes. Malleus, incus, stapes. Okay, so that's the sound hits the tympanic membrane, vibrates these bones, the malleus, the incus, and the stapes, and then those vibrate. The stapes ends up sending vibrations into the inner ear. Now, the inner ear contains semicircular canals, which are here, and then the vestibule and the cochlea, which looks like a snail. And then you have two nerves that kind of merge together, but we have the vestibular nerve and the cochlear nerve, which we call it, together we call it the vestibulocochlear nerve. But the auditory tube, this is the where sometimes person will have to get tubes in their ears to drain that. So let's see. Let's go back. All right. So the outer ear contains the pinna and the auditory canal. And the pinna is just the flappy part of your ear. And then the middle ear begins with the eardrum or tympanic membrane and ends at the oval and round windows. So it contains the ossicles or bones, and those bones are called the malleus or hammer, the incus or anvil, and the stapes or the stirrup. The auditory tube, which is also called the eustachian tube, extends from the middle ear to the nasopharynx, and it permits equalization of air pressure. You may have noticed if you ever traveled in the higher elevations in the mountains that your ears popped, and you could you could hold your nose and kind of blow out while you're holding your nose, and that can sometimes cause your ears to pop, and you'll feel better. You'll notice the pressure, getting better in your ears and your head. So, but that's the eustachian tube or the auditory tube. Okay, so the inner ear has, and you've got to remember that the ear is not just for hearing, it's also for equilibrium. So the areas that deal with equilibrium are the semicircular canals and the vestibule, and then the cochlea is responsible for hearing. So the semicircular canals and the vestibule are going to be for equilibrium, and the cochlea is going to be for hearing. And we'll talk about both, but just real quick, inner ear again, semicircular canals, and then here's the vestibule, and those all deal with equilibrium. And then the inner ear also has the cochlea, which deals with hearing. All right. And it said the oval window and the round window. Where are those? Round window is here, and the oval window is somewhere. There's the round. It's going to probably be on the inside. Okay, we'll see in a minute. All right. In the inner ear, we'll start with hearing first. Okay, so the spiral organ, which also is known as the organ of Corti, contains hair cells that synapse with the cochlear nerve, which is basically the auditory nerve. It's the portion of the vestibulocochlear nerve that deals with hearing. Okay, so synapses with the cochlear nerve or auditory nerve, and the hair cells are actually mechanoreceptors that are stimulated by sound waves. So they're mechanoreceptors for sound. That means that different sounds will bend those hair cells. But the bending of the hair cells will cause the basilar membrane to vibrate. And we'll see that, we'll see these structures in a minute. The stereocilia of the hair cells will bend. Okay, so the sound will vibrate the basilar membrane, and then that will cause the bending of the stereocilia of the hair cells. And then the nerve impulse will be transmitted to the cochlear nerve, and it will travel from the cochlear nerve to the brain and then to the auditory areas of the cerebral cortex, which really are in the temporal lobe. Okay, so if you look here, this is the cochlea, right? So sound waves are transmitted. They vibrate the malleus, incus, and stapes, and then the stapes vibrates against the basilar membrane, which is in this area here. And then the vibration of that basilar membrane, which is right there, okay, vibration of that basilar membrane vibrates or bends stereocilia in the hair cells, and that causes the nerve impulse to be transmitted through the cochlear nerve to the temporal lobe of the brain. Now, for equilibrium, that is controlled by these statocysts, which are gravitational equilibrium organs. And, let's go to humans. All right, here we go. All right. We have something otoliths, but not statocysts. If we do, I don't see it on here. Okay, so forget that. Just in humans, we have what's called rotational equilibrium, which involves the semicircular canals. Our hair cells have stereocilia embedded in a gelatinous cupola in the ampulla. Oh my gosh. Okay, so basically there's this gelatinous fluid, and the stereocilia from the hair cells are embedded in this gelatinous fluid within those semicircular canals, within the base of those semicircular canals. And so when we move our head, anytime we move our head, the stereocilia will bend. So head movements will cause those stereocilia to bend and send impulses to the vestibular nerve. All right. And then for gravitational equilibrium, we have mechanoreceptors for gravitational equilibrium in the utricle and the saccule. All right, so your semicircular canals are here. The utricle and the saccule are there and there. So like here and here. So semicircular canals are for rotational equilibrium. The utricle and the saccule are here and here, and they are for gravitational equilibrium. All right, so they detect movements of the head in any direction and help maintain gravitational equilibrium. Basically, again, you have stereocilia from the hair cells that are embedded in a gelatinous material called the otolithic membrane, and movements of the head cause sagging of that membrane, and that will bend those stereocilia. So sagging of the membrane bends the stereocilia, and then the nerve impulse is transmitted to the vestibular nerve. All right, so the semicircular canals have, at the base of each of those semicircular canals, you have endolymph, which is gelatinous material, and the stereocilia of the hair cells extends into that endolymph, and movement of your head. Notice he's rotating his head, so this is rotational equilibrium. Rotation of your head will bend the stereocilia, and then the nerve impulse is sent to the vestibular nerve. And when for gravitational equilibrium, that is detected. Still, you have this gelatinous material within the otolithic membrane, this is within the utricle and the saccule, here and here, and movements like this lady is nodding her head forward, that type of movement, this is for gravitational equilibrium. So that type of movement is going to bend the stereocilia of the hair cells, which will transmit those impulses to the vestibular nerve. So disorders of hearing and equilibrium, you can have tinnitus, which is ringing in the ears. You can have deafness. Certain types of deafness can be treated with cochlear implants and certain types cannot, and there are different degrees of deafness. There's total and partial. You can have vertigo, which is an equilibrium disorder. It's a feeling that the person is moving or their environment is moving, even though there's no movement. Then there's Meniere's disease, which is characterized by several of the above: vertigo, tinnitus, and hearing loss, some partial hearing loss, and it can even lead to complete hearing loss. Okay, our last section is on the somatic senses, and I'll do another. In fact, I'll probably just cover this in class, so I won't cover this in the recording. I'll just stop with section 4.